Good morning, everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I hold a PhD in this field. Today, we'll be exploring the qualifications of HPLC columns. This is the first slide, so I want to start by giving you a brief overview of what we'll cover. We'll discuss the key aspects that determine the quality and performance of HPLC columns, which are essential for accurate and reliable chromatographic analysis. I'm excited to share this information with you, so let's get started. Let's start by talking about what HPLC column qualification really means. Essentially, it's about making sure that the column we're using is suitable for the specific analytical purposes we have in mind. This step is crucial because it directly impacts the accuracy and reliability of our data. When the column is properly qualified, we can trust that our results are consistent and reproducible across different runs. Now, why is this so important? Well, for one, it plays a key role in maintaining data integrity. If the column isn't performing as expected, or if it's not suitable for the method, our results could be compromised. Proper qualification helps us avoid those issues by confirming that the column performs to the required standards. This is also tied to regulatory expectations. Agencies like ICH and USB have clear guidelines that emphasize the importance of system suitability and column performance validation. For example, ICH Q2, R1, outlines validation requirements to ensure our methods are reliable. Similarly, USP 621 specifically addresses chromatography system suitability and column performance testing. In essence, column qualification isn't just a good practice. It's a regulatory requirement that helps us ensure high-quality, reproducible results in our analytical work. It ties directly into the overall goal of delivering accurate data that stands up to scrutiny and regulatory review. Let's talk about why column qualification is so important in our analytical processes. First, it helps prevent analytical errors, which can lead to inaccurate results. When we properly qualify our columns, we also minimize retention time shifts, ensuring our measurements stay consistent over time. Poor resolution is another issue that column qualification can address, helping us separate compounds more effectively. This all ties into supporting key activities like method validation, where we need to confirm our methods are reliable, and routine quality control, where consistency is critical. Additionally, proper qualification is essential for regulatory compliance, making sure we meet all industry standards. Ultimately, by thoroughly qualifying our columns, we can reduce batch rejections, saving time and resources, and maintaining the integrity of our analytical work. Let's take a closer look at the regulatory framework that guides our analytical procedures. First, we have the ICH guidelines, which are internationally recognized standards. Specifically, Q2, R1, focuses on the validation of analytical procedures, ensuring these methods are reliable and reproducible. Then there's Q14, which emphasizes the development of analytical procedures right from the start. Moving on, we also reference USB standards, which include specific tests like column performance tests to ensure our chromatography columns are functioning properly. The FDA guidance provides clear expectations for the robustness of analytical methods, making sure they can withstand variability and still produce accurate results. Similarly, EMA and other regional authorities have comparable expectations, especially regarding documented verification. This means that regardless of where we operate, the regulatory bodies want to see thorough, documented proof that our methods are verified and fit for purpose. Overall, these guidelines form a cohesive framework that underpins the quality and reliability of our analytical work, aligning with our broader goal of ensuring product safety and efficacy. Now, let's take a closer look at the scope of column qualification. This process applies to several key situations. First, it covers new columns that are being introduced into the system. Second, it includes columns that have been stored or unused for a long period, especially after they've been sitting idle. Third, if columns are moved between different instruments, they also fall under this scope. However, it's important to clarify what is not included. Routine method suitability tests, for example, are not considered full qualification. These are more about ensuring the method works as expected in general, rather than qualifying the column itself. Understanding these distinctions helps us focus our efforts where they truly matter in maintaining quality and consistency in our processes. Now, let's walk through the typical workflow involved in column qualification. First, we start with a thorough documentation review, making sure all the specifications and previous data are in order. Next, we conduct a visual inspection to check for any physical damage or irregularities. Then, system suitability checks are performed to ensure the system is capable of producing reliable results. Following that, test sample injections are carried out to evaluate the column's performance. We then assess the performance to verify it meets our expected standards. Finally, we verify the acceptance criteria to confirm the column is qualified for routine use. Moving on to the key parameters we consider during column qualification, the first is column identity. This involves confirming the column type, manufacturer, and part number to ensure we're working with the correct equipment. We also look at the dimensions, specifically the length, internal diameter, and particle size, since these influence separation performance. The stationary phase chemistry is another critical factor, whether it C18, C8, phenyl, or other types, as this affects the selectivity of the separation. Lastly, physical integrity is essential. We need to ensure there are no blockages and that the pressure remains consistent throughout the operation. All these parameters together help us confirm that the column is suitable for our analytical needs and maintains the quality and reliability of our results. Now let's move on to slide 7, which covers system suitability tests, or SST. These tests are a crucial part of ensuring our method is performing reliably before we start analyzing actual samples. We begin by injecting a standard mix, which serves as our benchmark to evaluate various key parameters. First, we look at the retention time, which helps us confirm that our analytes are eluting consistently at the expected times. Next, we assess the theoretical plates, often denoted as N, to gauge the efficiency of our chromatographic system. Higher values generally mean better separation. We also evaluate the tailing factor, which indicates the symmetry of our peaks. Ideally, we want minimal tailing for accurate quantitation. Another important aspect is resolution, especially between critical pairs, to ensure that compounds are well separated and not overlapping. All these evaluations are done based on acceptance criteria derived from our validated method, ensuring that everything is within the acceptable range before proceeding. In essence, SSD helps us confirm that our system is suitable and performing consistently, which is vital for generating reliable and reproducible results throughout our analysis process. Now, let's take a closer look at performance parameters and acceptance criteria. These are crucial because they define the standards we need to meet to ensure our project's success. Performance parameters refer to the specific metrics and benchmarks that measure how well a system or component functions. Think of them as the goals we set to gauge performance, such as speed, efficiency, or reliability. Acceptance criteria, on the other hand, are the conditions that must be satisfied for the project or a particular deliverable to be considered complete and acceptable. They act as checkpoints, ensuring everything aligns with our expectations and requirements. By clearly defining these points early on, we create a shared understanding of what success looks like. This not only helps us stay focused, but also makes the evaluation process more straightforward. Connecting this to our overall theme, setting clear performance parameters and acceptance criteria ensures quality control throughout the project. It helps us avoid surprises later and promotes accountability within the team. Remember, clarity here leads to smoother execution and better outcomes in the end. Now, let's talk about requalification and periodic checks for our columns. First, it's important to know when to requalify. Requalification should be done after any column storage, especially if the column has been idle for a while. Also, after any misuse or mishandling, it's crucial to reassess the column's condition. Additionally, if there's a change in method parameters, requalification ensures everything still performs as expected. On the other hand, periodic checks are just as vital. Typically, these are performed monthly or weekly, depending on how frequently the column is used. Regular checks help us maintain trending data, so we can spot any performance issues early. Moving on to column fitness for purpose. This is about demonstrating that the column continues to meet our performance criteria. It should reliably produce accurate results, ensuring data traceability. This includes tracking batches or lots of the column, along with instrument and analyst details, so we have full traceability and accountability. In essence, both requalification and regular checks are about ensuring the longevity and reliability of our columns, ultimately supporting the integrity of our analytical results. Let's talk about troubleshooting column issues and some best practices to keep things running smoothly. First, it's important to equilibrate your column before use. This helps stabilize the system and ensures consistent results. If you're working with complex samples or suspect contamination, using guard columns can be very helpful. They act as a protective barrier, extending the life of your main column. Documenting your mobile phase preparation is also key. Keeping detailed records helps track any changes that might affect performance. Additionally, maintaining instrument logs allows you to monitor trends and identify potential problems early. Training your analysts on proper qualification steps is essential. Well-trained personnel can recognize issues before they escalate. Now, when symptoms arise, understanding their causes can save you time. For example, a shift in retention time often points to mobile phase issues or column aging. Poor resolution might indicate deterioration of the column packing, and high back pressure usually suggests a blockage or particulate buildup. By understanding these symptoms and causes, and following best practices, you can troubleshoot more effectively and keep your chromatography system running at its best. Let's take a closer look at qualification documentation. This is a critical part of ensuring that our equipment and processes meet all necessary standards. Starting with the column qualification protocol, this document outlines the specific objectives, scope, and references that guide the qualification process. It's essentially our roadmap to make sure everything is covered thoroughly. Next, we have the test methods and acceptance criteria, which define how we evaluate performance and what standards must be met for acceptance. This helps us maintain consistency and quality across all qualifications. Then, we record the results and deviations. It's important to document what we find, including any deviations from expected outcomes, so we can understand the performance and address issues proactively. Following that, we draw a conclusion and determine the qualification status based on the results. This confirms whether the equipment or system is qualified and ready for use. And finally, it's essential to retain all these records in accordance with GMP and GLP requirements. Proper documentation ensures traceability, compliance, and readiness for audits. All these steps together help us maintain rigorous quality standards and validate that our processes are reliable and compliant. Let's wrap up with some key takeaways. Column qualification is absolutely critical across several areas. First, it directly impacts data quality, ensuring that the data we rely on is accurate and trustworthy. Second, it's essential for regulatory compliance, helping us meet the expectations of agencies like the FDA and other authorities. Third, it plays a vital role in method robustness, making sure our analytical methods are reliable and consistent. Now, to achieve these goals, it's important that the qualification process is well documented, reproducible, and risk-based. This means we should have clear records, procedures that others can follow and reproduce, and a focus on understanding and mitigating potential risks. Lastly, all of this must align with the expectations set by ICH, USB, and FDA. Following these guidelines ensures we're not only compliant, but also maintaining the highest standards in our work. So, in essence, thorough column qualification underpins the integrity of our data, regulatory adherence, and overall method reliability, which are all crucial for success in our field. For further support, please don't hesitate to reach out to us. We're here to help and answer any questions you might have. Feel free to contact us through the methods provided. And with that, I want to sincerely thank you for your time and attention today. Your engagement is truly appreciated, and I hope you found the presentation valuable. Remember, we're always here to support your success. Thank you once again.